We are going to return to the conversation about ATP, how we derive ATP, that is to say, how ATP is synthesized, sources of ATP, sources of glucose. Glucose is a huge source of fuel to make ATP, as are a number of the other macromolecules like lipids and proteins, specifically amino acids and triglycerides. We're going to delve into gluconeogenesis, which is the formation of glucose from non-carbohydrate precursors. We'll talk about glycolysis in more detail than we have previously, and let's just get going on that. Okay, so sources of ATP. There's going to be some residual ATP within our cells that can be used up immediately upon exercise. And really what we're talking about here, what I'm trying to elaborate on here, is muscle metabolism. That is to say, the breakdown of macromolecules to produce ATP to fuel our muscles or energize our muscles so they can do work. So we have the residual ATP in the muscle cells that's going to be used up very quickly. There is a molecule known as creatine phosphate. It's also known as phosphocreatine, which is the exact same thing. And this is a molecule, creatine that is, that binds an inorganic phosphate. It can hold on to the inorganic phosphate until more ATP is needed to be made. So that phosphate that's attached to creatine will combine with ADP. So ADP plus a phosphate will make ATP. And that is a quick source of producing ATP during muscle activity. The creatine phosphate is only going to sustain muscle for about 15 seconds. And then the other source of ATP is the glycolysis citric acid cycle, the electron transport chain. Citric acid cycle, once again, is the same thing as the Krebs cycle. And when I say electron transport chain, I am including chemiosmosis and oxidative phosphorylation within that equation. To be clear, the body, our cells, our muscles can use fats to produce ATP. And as a matter of fact, during low intensity exercise, most of our ATP is coming from fats. But during an oxygen deficit, there is a decreased metabolism in fatty acids, and we'll talk about that as we move forward. Okay, let's just review some sources of glucose that will be used to make ATP. Our blood plasma contains glucose, and that is our first primary source of glucose to make ATP. Glycogen is the storage form of glucose. We find glycogen primarily in muscle cells and in the liver. Glycogen can be broken down via the process of glycogenolysis to put more glucose into the blood to increase our blood glucose levels. And then gluconeogenesis is the production of glucose from non-carbohydrate precursors, which we'll discuss in detail. To be clear, after 30 minutes of aerobic exercise, fatty acids are released into the bloodstream and metabolized to produce ATP, though they are metabolized at a much slower rate than glucose is. That is to say, it takes much longer to produce ATP via fatty acids than via glucose. Okay, so let's take a more in-depth view of glycolysis. We've discussed glycolysis in previous videos, but we're going to do so with more detail here. Once again, the precursor to glycolysis is glucose. Glycolysis always starts with a molecule of glucose, C6H12O6, and glycolysis ends with the molecule pyruvate. Pyruvate is a 3-carbon molecule. Glucose is a 6-carbon molecule. So there are 2 pyruvate molecules produced via the process of glycolysis. 2 3-carbon molecules of pyruvate, total 6 carbons, and those were the 6 carbons from the original glucose molecule. The first step in glycolysis, keep in mind, glycolysis is a 10-step enzymatic process. That is to say, there is an enzyme required for each of the steps within glycolysis. I'm not really concerned about the enzymes for glycolysis, with the exception of the first step of glycolysis, which we see right here. The enzyme utilized for the first step in glycolysis is hexokinase. And hexokinase converts glucose to glucose 6-phosphate. And it does so by breaking down ATP. And this is one of the many important steps of glycolysis. That is to say, there are 2 steps in glycolysis where ATP is going to be used. If we recall to previous lectures, glycolysis produces a total of 4 ATP, but it also uses 2 ATP via the entire process. So we tend to say, or I tend to say, glycolysis produces a net 2 ATP. That is because glycolysis is using ATP in a couple of the steps. And the first one we see right here, which is the conversion of glucose to glucose 6-phosphate, where we get the phosphate added to glucose from ATP. The second step just changes forms. It's an isomer of glucose 6-phosphate, changing glucose 6-phosphate into fructose 6-phosphate. And then step 3, we see ATP being used again. ATP is broken down to liberate the phosphate to add it to fructose 6-phosphate, converting fructose 6-phosphate into fructose 1,6-biphosphate. And to be clear, these numbers, the 6 and the 1 and the 6, depending on which molecule we're looking at, is designating which carbon the phosphates are bound to. I'm not overly concerned with that, but that's the derivation of these numbers. So step 3 converts fructose 6-phosphate into fructose 1,6-biphosphate via the hydrolysis of ATP, liberating one phosphate, adding it to that molecule. Okay, what we see right here is the conversion of fructose 1,6-biphosphate into glyceraldehyde 3-phosphate. Now, we see this 5 over here to the right, and that's because glyceraldehyde 3-phosphate changes forms from glyceraldehyde 3-phosphate to dihydroxyacetone phosphate, otherwise known as DHAP. Glyceraldehyde 3-phosphate is the isomer that is going to proceed through glycolysis. It is not the DHAP molecule, but that's why I have the 5 over here, because the 5th step is where it changes forms from glyceraldehyde 3-phosphate to dihydroxyacetone phosphate back and forth. But the important thing I want you to remember here is fructose 1,6-biphosphate is broken down into 2 molecules of glyceraldehyde 3-phosphate, which will proceed through the process of glycolysis. So now we are dealing with 2 molecules of glyceraldehyde 3-phosphate. And so once again, I can't stress this enough. Every molecule we're looking at from here forward within glycolysis, there are going to be 2 of them, and that's why I have the 2x preceding all of these subsequent molecules. Glyceraldehyde 3-phosphate in step 6 is going to be converted to 1,3-biphosphoglycerate. And in that process, 2 NADH molecules are going to be produced. The 2 NADH and the 2 protons that we see right here. Keep in mind, as I've stressed before, with glycolysis and with the subsequent Krebs cycle, arguably NADH and FADH2 are much more valuable than the ATP that are produced via these processes, because they will culminate in producing many, many more ATP via the electron transport chain, chemiosmosis, oxidative phosphorylation that occurs later within the inner mitochondrial membrane. So this process, or this step 6, will result in phosphorylating glyceraldehyde 3-phosphate to produce 1,3-biphosphoglycerate. Moving on to step 7. This is the first step where ATP is being produced. The conversion of 1,3-biphosphoglycerate into 3-phosphoglycerate is going to result in the production of ATP. And the reason why it produces 2 ATP molecules is because we have 2 molecules of 1,3-biphosphoglycerate being converted into 2 molecules of 2-phosphoglycerate. So to be clear, we see 1,3 in biphosphoglycerate and only 3 in phosphoglycerate because it has lost a phosphate. And those 2 phosphates have been added to 2 ADPs to produce 2 ATP molecules. That 3 phosphoglycerate in step 8 is going to be converted to 2 phosphoglycerate. And then 2 phosphoglycerate in step 9 will be converted to phosphophenylpyruvate. There are going to be 2 phosphophenylpyruvate, just like there were 2 phosphoglycerate and 2 3-phosphoglycerate molecules, so on and so forth. And then here in step 10, we see phosphophenylpyruvate being converted to pyruvate. Phosphophenylpyruvate is going to lose its phosphate. I should say both those molecules of phosphophenylpyruvate will lose each of their phosphates that will be added to ADP. I realize right here this says 2 ATP. That should say 2 ADP being converted to 2 ATP. So as we see here in step 7 and step 10, there are 2 ATP molecules produced in each of those steps, totaling 4 ATP. But we use 2 ATP in step 1 and in step 3. Hence, there are 2 net ATP molecules produced via the process of glycolysis that ends up with the production of 2 pyruvate molecules. Okay, so let's just talk quickly about oxygen deficiency. That is to say, how do we get ATP during oxygen deficiency? The first thing to understand is that while we talked about fatty acid metabolism to produce ATP, which we will detail shortly, there is a decrease in fatty acid metabolism in the absence of oxygen. So the options are using some of the residual ATP that we talked about. The other options are anaerobic glycolysis, that is to say the production of ATP via glycolysis in the absence of oxygen. And to reiterate what we've discussed many times, glycolysis does not require oxygen. Glycolysis occurs in the presence or absence of oxygen. So in the absence of oxygen, glycolysis is going to proceed just like we outlined. That is to say, it's going to produce the 2 net ATP. So during anaerobic glycolysis, during oxygen deficiency, glycolysis will proceed as long as glucose is available or some substrate is available to produce the pyruvate. Pyruvate in the absence of oxygen is going to be converted to lactate. Here we see lactate being produced from pyruvate in the absence of oxygen. Lactate serves as somewhat of a reservoir to consume pyruvate. And it's not getting rid of pyruvate, it's really just hiding pyruvate. Because if pyruvate did not convert into lactate, there'd be an increasing concentration of pyruvate within the cell. The increasing levels of pyruvate concentration would shut off glycolysis. Glycolysis, or any metabolic process, is only going to proceed as if the end product is in low concentrations. So with pyruvate being converted to lactate, that maintains low levels of pyruvate concentrations within the cell. So that is one huge function of lactate. Lactate in the presence of oxygen, that is to say, if oxygen was not present, pyruvate turns into lactate. Once oxygen is present, lactate can be reconverted back into pyruvate, at which point it can then enter the Krebs cycle. Not directly, it's going to react with coenzyme A to create acetylcoenzyme A, and then eventually enter the Krebs cycle, and we will outline that as well. And pyruvate can also be reconverted back into glucose. Let's just discuss what happens during hypoxia. That is to say, there's absolutely no breathing going on. There is going to be some residual oxygen in the lungs, and there is going to be anaerobic glycolysis that continues because of the oxygen that had remained bound to hemoglobin. So let's just say someone stops breathing. There's going to be some residual hemoglobin or residual oxygen bound to hemoglobin. and there's going to be some oxygen in the lungs. Okay, so let's talk about gluconeogenesis. Once again, gluconeogenesis is the production of glucose from non-carbohydrate precursors. To be clear, glucose is the primary energy substrate for the brain and the red blood cells. But there still needs to be glucose present between meals. So if we eat a huge meal, there's generally going to be sufficient glucose available to make ATP. In the absence of adequate levels of glucose, the body can engage in glycogenolysis, which is not to be confused with gluconeogenesis. Glycogenolysis is breaking down storage form of glucose in muscle and in the liver. That is to say, glycogenolysis breaks down glycogen and puts glucose into the blood. Glycogen is the storage form of glucose. There is a litany of glucose molecules that make up glycogen. When glycogen is broken down, it increases glucose levels in the blood. Gluconeogenesis is the synthesis of glucose, mainly from lactate, which we just previously outlined, and amino acids, and to a certain degree from triglycerides, which we see up at the top right here. To be very clear, for amino acids, only 60% of amino acids can be converted into glucose. And this is done via the process of deamination, which I will show you a slide of that. And then triglycerides can be broken down into glycerol and the fatty acid side chains. The glycerol will enter glycolysis via an intermediate stage, and the fatty acids will be broken down into ketone bodies. This image right here is showing glycerol in the upper left-hand corner here. Glycerol, when it reacts with ATP, is going to make a molecule known as glycerol 3-phosphate, which will then enter at roughly step 5 in glycolysis. Now, to be clear, glycerol 3-phosphate is going to be converted directly into dihydroxyacetone phosphate, which I mentioned earlier, which in step 5 fluctuates between DHAP and glyceraldehyde 3-phosphate, which is the form that proceeds down to step 6. So glycerol will be converted ultimately into glyceraldehyde 3-phosphate and then proceed. Keep in mind, we are only dealing with one molecule of glycerol, hence one molecule of glyceraldehyde 3-phosphate, not two that would have been created from glucose. And to be clear with the fatty acids, once again, triglycerides are composed of a glycerol molecule and then 3 fatty acid side chains. The fatty acids will be broken down via a process known as beta oxidation. Beta oxidation breaks down the fatty acid side chains into 2 carbon fragments. The 2 carbon fragments will be converted to acetyl coenzyme A, and as we know, acetyl coenzyme A gets converted or enters the Krebs cycle. If acetyl coenzyme A from lipolysis, and once again, lipolysis is the breakdown of triglycerides or any lipids for that matter. If acetyl coenzyme A from lipolysis is produced faster than can be used by the Krebs cycle, ketone bodies are produced. Ketone bodies are a main source of fuel for the brain during starvation. Now, this certainly could lead to a condition known as ketoacidosis, which is not necessarily good, but ketone bodies do serve their purpose to fuel the brain during times of starvation. Let's look at deamination. That is to say, the process that happens en route to producing glucose via amino acids. Amino acids are the building block of proteins, and amino acids are composed of 3 different parts. An amino group that we see to the left, which is based upon the atom of nitrogen, the carboxyl group over to the right, and the side chain represented by R right here. Each amino acid differs from each other based on the side chain. So anything other than the amino group is the carbon skeleton. That is to say, all of this makes up the carbon skeleton. Deamination gets rid of the amino group from the amino acids. And once again, only 60% of all the 20 amino acids in our body can be used to make glucose. Deamination is going to cleave the bond between the amino group and the remaining carbon skeleton, the side chain and the carboxyl group. And here, the amino group will be converted into ammonia. Ammonia is toxic to the body. And then that ammonia will be converted to urea or sometimes uric acid and will be eliminated from the body via urine. Or that ammonia can be converted to another amino acid via the process of transamination. The carbon skeleton is going to be converted to glucose or to acetyl coenzyme A or to ketone bodies. Glucose can proceed through glycolysis. Acetyl coenzyme A can ultimately enter the Krebs cycle, and the ketone bodies can serve the brain in times of need. So I keep talking about acetyl coenzyme A and pyruvate. So I just want to review the role of pyruvate in acetyl coenzyme A. In the presence of oxygen, pyruvate will combine with coenzyme A to produce acetyl coenzyme A. That process is going to result in the production of carbon dioxide and some NADH. But what I'm concerned about in this image right here is in the presence of oxygen, pyruvate reacts with coenzyme A to produce acetyl coenzyme A. That acetyl coenzyme A is broken down into an acyl unit, and that acyl unit is going to enter the Krebs cycle, otherwise known as the citric acid cycle. It is not acetyl coenzyme A that enters the citric acid cycle, it's the acyl unit. And the acyl unit will combine or react with the end product of this cycle, the end product of the Krebs cycle, which is known as oxaloacetate. Oxaloacetate is a 4-carbon molecule. It is reacting with the acyl unit, which is a 2-carbon molecule. Oxaloacetate reacting with that 2-carbon molecule is going to produce citrate, and then that's going to proceed through the citric acid cycle, making some ATP, but primarily making NADH and FADH2, which will shuttle electrons to the electron transport chain. Because it is a cycle, it's always going to end up producing oxaloacetate, which, provided any more of the acyl unit, will produce more citrate, and this process or cycle will continue. One thing I want to point out is acetyl coenzyme A, like pyruvate, is a 3-carbon molecule. It breaks down into the acyl unit, which is a 2-carbon molecule, which leaves us one more carbon, and that is this coenzyme A. That is to say, acetyl coenzyme A is broken down into that acyl unit and a coenzyme A. And that coenzyme A will be recycled back to react with another molecule of pyruvate. Okay, let's talk about regulation of gluconeogenesis. The first stimulus to initiate the process of gluconeogenesis is a decreased plasma glucose concentration, diminished levels of glucose within the blood. The second stimulus for gluconeogenesis is cortisol. Cortisol is released from the adrenal cortex, and via this stimulus from CRH from the hypothalamus and ACTH from the anterior pituitary, the adrenal cortex releases cortisol. Cortisol releases proteins from cells, putting the proteins in the blood. The proteins are broken down, and the amino acids are used to produce glucose. That is to say, that process we just outlined before via deamination, amino acids are going to be converted to glucose. So cortisol plays a large role in the process of gluconeogenesis.